There are two or three important research that has come in this meeting that I think it will get into the clinical arena pretty soon. First of all, in the gene area, there is these studies of titin truncation in dilated cardiomyopathy that very likely the clinicians will have to start looking at when we assess our patients, because this is a treatable genetic disorder. So this is very important. We had two very good abstracts in the meeting. Second of all, how do we manipulate stem cells before we implant them? There are interesting epigenetic and physiological treatments that we can perform before we put the stem cells into the body, into the heart, to reduce myocardial infarction. And the third of all is there are new improvements in tissue engineering, there are new constructs, there are new cells, and there is more and more knowledge on how iPS-derived cardiomyocytes are going to be used in conjunction with scaffolds to be applied in the areas of infarction to reduce ventricular remodeling.
We traditionally are very restrictive in our approach of understanding disease, and the abstract I presented really addresses the crosstalk between different cell types in determining the phenotype of cardiac function. And this specifically addresses the crosstalk between two cell types that are very rarely considered in cardiac science, which is the interaction between red blood cells and cardiac myocytes. Red blood cells may produce reactive oxygen species that then are influencing the function of the cardiac myocyte in the setting of ischemia reperfusion. We have been able to isolate venous red blood cells and to directly measure nitric oxide and superoxide anion from these red blood cells. So this opens the possibility to really, just through a venopuncture, measure how risk factors impact on the production of ROS from circulating red blood cells.
Extracellular RNA was a big discovery. It seems like there's a lot of RNA in cells, and when heart muscle is damaged after a heart attack, a massive amount of this RNA is released into the tissue. And this activates inflammation strongly by a completely novel mechanism. But the good news is that we already have drugs that can target this mechanism. And so it implies that fairly quickly that this could be translated into a benefit for patients.